Good morning, students. So, in last class, we have discussed the excretory products and their elimination. So, there are three major excretory products are presenting. One is aminotelic, ureotelic, and uricotelic organisms. If the organisms which excrete the main excretory product in the form of ammonia, we can call them as aminotelic organisms. Here examples are invertebrates and also the tadpole larva of the frog. And ureotelic, the organisms which excrete the main excretory products in the form of urea are called as ureotelic. This is less toxic when compared to the ammonia. So here the cartilaginous fishes, the frog as well as humans, they excrete the mainly excretory product in the form of an urea. Next, the third one is uricotelic. If the organisms excrete the main excretory product in the form of a uric acid, then we can call them as the uricotelic organisms. Here example, we can take in place reptiles, the birds and insects where they excrete the mainly excretory product in the form of a uric acid. So it is when compared to the uric acid, here ammonia is highly toxic. When compared to the urea, it is less toxic, and here very less toxic is the uric acid. So here for elimination of uric acid, here requires only a 10 ml of water is required to eliminate 1 gram of uric acid. But in ammonia, it requires 300 to 500 ml of water is required to eliminate 1 gram of ammonia. In uricotelic organisms, that means for urea, here required 50 ml of water is required to eliminate the urea. So here ammonia is highly toxic when compared to the urea, and when compared to the uric acid, urea is highly less toxic. So this is where we have studied, and also we have studied the structure of the kidney. In a kidneys, here we are having a pair of ureters, a pair of kidneys and ureters, and urinary bladder and urethra is presenting, where here these are presenting, a pair of kidneys are present in the pelvic regions, and these will consist of a pyramid like structures. 15 to 16 pyramid like structures are called as renal pyramids. From the renal pyramids will having a renal papillae. Through the renal papillae, the urine is allows and enters into the pelvis, that is hilum, hilus to the pelvis, renal pelvis. From renal pelvis it passes to the ureter. From ureter it passes to the urinary bladder. Through urethra it is excreted and outside. So in females the urethra is taken place in the form of urethral orifice. But in males, here the urethra is combined with a genital system where it is called as a urogenital system. That means urine as well as genital products are semen is produced from the same duct through the urethra. It enters through the penile regions, through the penis. At the tip of the opening of penis, it excreted and outside or exit outside of the body. So here in males we can call that as a urogenital system. And also in kidney, each kidney will consist of 1.5 uniferous tubules are presenting. That uniferous tubules are also called as the nephrons. So nephrons are the structural and functional units of kidneys. Now coming to the structure of the nephron. So here nephrons in each kidney, will consist of 1.5 millions of the nephrons are presenting. Where the nephrons are these are the structural and functional units of kidneys. These are the structural and functional units of kidneys, where that is called as the nephrons. So here nephrons are the structural and functional units of kidneys, where here each kidney will consist of 1.5 millions of nephrons are presented. And each kidney, each nephron will consist of different parts. That means each nephron consists of different parts, it consists of different parts are presenting, such as first one we can call as the Malpighian tubule. First one we can call as a Malpighian tubule. Second one is called as a proximal convoluted tubule, and the third one is the Henle's loop, and the fourth one is a distal convoluted tubule, and the fifth one is a collecting duct, and lastly that is called as a vas recta, or otherwise you can call as a peritubular capillaries. This is where we can take in place. These are the different regions. Where they are presenting in the nephron. One is a Malpighian tubule, second one is a proximal convoluted tubule, a Henle's loop, a distal convoluted tubule, a collecting duct, as well as the peritubular capillaries. So this is where the different parts which are present in the nephron. Now we will discuss the one by one, we will discuss detailedly, that is each part will be discussed, that is one by one. So if you see in this is so now coming to this diagram of the nephron, I will show that. Now into the diagram of the nephron, this is called as glomerulus, this is called as Bowman's capsule, and this is called as PCT. PCT means proximal convoluted tubule, and this is called as the Henle's loop. So in Henle's loop it is consists of the descending limb, and this is an ascending limb, and this is called as a DCT. DCT means a distal convoluted tubule, and this is called as the collecting duct. So here from the collecting duct will consist of the peritubular capillaries are presenting, which are running parallel to the one another. Which are running parallel to the one another. We can call them as a peritubular capillaries. So these are the different regions which are presenting in the nephrons, that is will having a Bowman's glomerulus and a Bowman's capsule. Together we can call as the Malpighian. It is called as a a Malpighian tubule. So here what is Malpighian tubule means? It is a combination of here a Malpighian tubule. It is a combination of a glomerulus as well as the Bowman's capsule. Together we can call as the Malpighian tubule. It was introduced by Marcello Malpighi. So instead of his name, we can call this as the Malpighian tubule. It was introduced by Marcello Malpighi. So instead of his name, it is called as the Malpighian tubules. And proximal convoluted tubule, Henle's loop, that is descending limb as well as ascending limb, and DCT, distal convoluted tubule, collecting duct, and also consists of the peritubular capillaries. Now coming to the different parts which are presenting, now we will discuss, firstly we will discuss the Malpighian tubule, later that is PCT, distal convoluted tubule. Likewise one by one will be discussed now. Now coming to the Malpighian tubule, the Malpighian tubule. So here in the Malpighian tubule, it is also called as a renal capsule, is also called as renal corpuscle. It is also called as renal corpuscle, renal corpuscle. It is also called as a renal corpuscle. It is also called as renal corpuscles. It is also called as a renal corpuscle. So here that it is called as renal capsule, where it is present in a renal cortex. It is present in a renal cortex. Here already we know that here kidney is differentiated into two regions. Outer region is called as a cortex and inner region is called as the medulla. So outer region, here this Malpighian tubule, the Malpighian tubule is presenting in the renal cortex, where in the Malpighian tubules here it consists of two parts are presenting. That two parts are, one is called as the glomerulus and the second one is called as the Bowman's capsule. So glomerulus plus Bowman's capsule together we can call as the Malpighian tubule. Here what is Malpighian tubule? It is a combination of the glomerulus as well as the Bowman's capsule. Together we can call as the Malpighian tubule. Why it is called as Malpighian means it was introduced by the Marcello Malpighi. So instead of his name we can call that as the Malpighian tubule. So here we are having glomerulus as well as the Bowman's capsule. So here what are glomerulus? Here what is the glomerulus? So here what is glomerulus means? These are a tuft of blood capillaries, a tuft of blood capillaries, a tuft of blood capillaries present in the Bowman's capsule. That means these are a tuft means a group of blood capillaries which are presenting in the cup-shaped structure in the glomerulus, that is in the Bowman's capsule, that is called as the glomerulus. This is arised from the renal arteriole. It is produced from the renal artery. It is produced from the renal artery, where it is differentiated into two regions. One region, that means the artery which enters into the kidney. The arteries which enters into the Bowman's capsule we can call as an efferent arteriole. What is efferent arteriole means? The blood vessels are the arteries which enters into the Bowman's capsule. So here renal artery will enter. The renal artery means it is a blood vessels which carries the oxygenated blood to the kidneys is called as renal artery. This renal artery will differentiate when enter into the kidney. It is differentiated into two regions. One is called as an afferent arteriole. It is a wide afferent and arteriole. It is a wide efferent arteriole and a narrow efferent, where here this narrow efferent arteriole will leaves the kidney, that is leaves the Bowman's capsule. That means the artery which enters into the Bowman's capsule is called as afferent arteriole, and the artery which leaves the Bowman's capsule we can call as the efferent and arteriole. So here a wider efferent arteriole to the a narrow efferent arteriole, the blood will enters, where here it helpful for the filtration of the blood. That means in this Bowman, in the glomerulus, when enters into the Bowman's capsule, here a filtration of blood is taken place. That means the blood which consists of the harmful substances or the excretory waste materials which are presenting that are essential or non-essential elements which are presenting that are filtered in the into the Bowman's capsule. So the tuft of blood capillaries which are present in the Bowman's capsule, we can call them as a glomerulus. It is called as a glomerulus, where it is an arteriole. It is an renal artery, where it is differentiated into a wide efferent arteriole to the and narrow efferent arterioles. Next, coming to the second one, that is a Bowman's capsule. Here coming to the second one, that is a Bowman's capsule. Here coming to the second one, that is a Bowman's capsule. Bowman's capsule. So here what is that? Bowman's means capsule means it is a cup-shaped structure. This cup-shaped, it is a cup-shaped structure which is presenting that is called as the Bowman's capsule. It is called as what is the Bowman's capsule means it is a cup-shaped structure. It is a cup-shaped or a bowl-shaped structure which is presenting that a bowl-shaped structure which is presenting, or a cup-shaped structure which is presenting, we can call that as the Bowman's capsule. So this Bowman's capsule was given by the name, it was introduced the name by the after William Bowman. It was introduced by the scientist called as William Bowman. It was introduced by the scientist William Bowman. So instead of his name, it is called as the Bowman's capsule. Why it is called as Bowman's capsule means it was introduced by the scientist called as William Bowman. So instead of his name, it is called as a Bowman's capsule, where here this Bowman capsule is differentiated into two regions. It is differentiated into two regions. The outer is called as a parietal layer. Parietal layer. The outer is called as the parietal layer, and the inner is called as an visceral layer. So outer layer is called as the parietal layer, and inner is called as the visceral layer. So here in between these outer and inner layer will having a capsular space is presenting. That is, it is presenting between the space present between the two layers is called as the capsular space. In between the two layers, that is the outer and inner layer, will consist of a space is presenting. The space is called as the capsular space. So, in between the outer and inner layer, that is outer layer is called as a parietal layer and inner layer is called as a visceral layer. So, outer layer will consist of the specialized, it consists of the cells, that cells are called as the podocytes. Outer layer will consist of cells are presenting, these cells are called as podocytes. These podocytes will consist of specialized cells are presenting, that specialized cells which are they are called as pedicels. So, here in these podocytes will consist of specialized cells are presenting in a podocytes. In a podocytes will having the specialized cells are presenting or present, they are called as pedicels. So, here these pedicels, in between the pedicels they are having in between the podocytes will having the space is presenting, the space are called as the split spores. These split spores are helpful for this filtration when the blood is filtered through these split pores, that is enters into the Bowman's, into the glomerulus, that is into the Bowman's capsule, from Bowman's capsule through the split pores that enters into the proximal convoluted tubules. So, here having the specialized cells are presenting, that specialized cells we can call them as the spores between the food process, that is the food processing, they are called as split pores, which helpful for the filtration. So, due to the filtration of these pores we can call them as the filtrate pores also, filtrate pores. These are also called as the filtrate pores. Why? Means they filtered the blood, that means what is the blood containing the essential or non-essential elements which are presenting that are filtered through these pores into the proximal convoluted tubule, so that it is called as the filtration pores. And here this outer, the visceral parietal layer is made up of the squamous and epithelial cells. It is made up of squamous and epithelial cells. Here the parietal layer is made up of the squamous, it is made up of squamous and epithelial cells, and inner layer which is containing the visceral layer will having specialized cells are presenting, that specialized cells are called as podocytes. In between the podocytes will consist of the split pores. These split pores are helpful for the filtration of the blood. So, they are also called as the split filtration, they are also called as filtrate spores also. Why? Means they helpful for filtration of the blood which is enters into the Bowman's capsule from a wide and efferent arteriole to the a narrow and efferent arteriole. Here the blood will be filtered, and that filtered is entered into the nephric filters, that is enters into the Bowman's capsule. From Bowman's capsule it passes to the proximal convoluted tubule, from proximal it passes to the Henle's loop, Henle's loop to the distal convoluted, from the distal it connects to the collecting duct. So, here each nephron is connected with a collecting duct where it collects the waste materials which are presented. From the collecting duct, that is passes through the urinary bladders. From urinary bladder that are enters through the urethra, they are excreted outside through the body. This is how the process is taken place. So, in the Bowman's capsule will having the visceral layer will having specialized cells called as podocytes. In the podocytes will consist of the specialized cells are presenting, they are called as pedicels, and in between the podocytes will having a split pores are presenting. These split pores are helpful for the filtration of the blood, so they are called as the filtrate pores also. They are also called as the filtrate pores, where these are the two regions where they are presenting in the Malphizian tubules. So, here Malphizian tubules will consist of the two parts. One is called as the glomerulus and the second one is called as the Bowman's capsule. So here Malpighian tubule together it is called as the Bowman's, that is Malpighian tubule means a renal corpuscle. It is a combination of the the glomerulus and a Bowman's capsule. So this is the part where we can call as a Bowman's. Why it is called as Bowman's capsule means it was introduced by the scientist William Bowman. So instead of his name here it is called as the Bowman's capsule. It is called as a Bowman's capsule. Next, coming to the second one, that is a proximal convoluted tubule. Now we will discuss the second one, that is the proximal convoluted tubule. That is the proximal convoluted tubule. Second one is the proximal convoluted tubule. So this proximal convoluted tubule, it is present immediate next to the Bowman's capsule. This region is called as a proximal convoluted tubule, where here this proximal convoluted tubule, it is 15 centimeters in length. It is 15 centimeters in length, and here the proximal convoluted tubule, it is made up of a cuboidal and epithelial cells. It is made up of a cuboidal. It is made up of cuboidal epithelial cells. It is made up of cuboidal epithelial cells with the brush borders. That means here it consists of the microvilli are presenting. So here they consisting of these are having a cuboidal. It is made up of a cuboidal and epithelial cells, where it consists of it is 15 centimeters in length. It is made up of a cuboidal and epithelial cells, where it is presenting in the renal cortex. It is present in the renal cortex. It is extending into the renal cortex. It is made up of, and it is made up of cuboidal epithelial cells with a brush border like structures are presenting. This brush border like structures will consist of the microvilli, where these microvilli are helpful for increasing the surface area of the absorption. It helpful for increasing the surface area, of absorption. It helpful for the increasing surface area of the absorption, and the proximal convoluted tubule is mainly meant for the absorption of essential elements. It helpful for the absorption of essential elements. It is absorption of the essential. It helpful for the absorption of an essential elements. So here, due to the presence of where in the proximal convoluted tubule all essential elements are absorbed. Here it helpful for the absorption of an essential elements. So here it consists of the brush borders with a cuboidal epithelial cells are presenting. So in brush borders will consist of the microvilli are presenting, where it increase the surface area of the absorption is taken place in the proximal convoluted tubule. And the main function of the proximal convoluted tubule means it helpful for the absorption of all essential elements are absorbed in the proximal convoluted tubule. In the proximal, it is meant for the absorption of all essential elements. All essential elements, glucose, amino acids, the sodium, potassium, all these essential elements which are required for the body that are absorbed in the proximal convoluted tubule only, where it consists of a microvilli. These microvilli are helpful for increasing the surface area of the absorption. So, such region it is called as a proximal convoluted tubule. Next, coming to the second one, the third one, that is a Henle's loop. So, here in a proximal convoluted tubule, here it helpful for mainly meant for the reabsorption of the essential element from the nephric filtrate. Here absorption not, here reabsorption of essential element from the nephric filtrate. That is the main function of a proximal convoluted tubule. Next, coming to the Henle's loop, it is the longest region. It is the longest region present in a nephron, present in a nephron. So, it is the longest region where it is present in the nephron, where it is extends, it is present in a renal medulla. It is present in a renal medulla. It is presenting in a renal medulla, where here it is a longest a part of the nephron, where it is a U-shaped structure. It is a U-shaped structure present in the nephron. That U-shaped structure which is present in nephron is called as that is called as a Henle's loop, where it is differentiated into two regions, that is called as the descending lymph, and the second one is the ascending lymph. So, here the descending lymph which is presenting, it is the starting part, the initial part of descending lymph. It is made up of with a cuboidal. It is made up of a cuboidal epithelial cells. It is made up of a cuboidal epithelial cells with which is a thick in. That means this initial part of the distal limb. This distal limb is a thick segment. It is a thick segment where it is made up of. It is a thick segment which is made up of a cuboidal and epithelial cells. And next to this, having a thin segment is presenting a thin segment, where this thin segment is made up of the squamous. It is made up of a squamous epithelial cells. That means here it is made up of two segments. One is the thick segment. Initial part is made up of a thick segment where it consists of cuboidal epithelial cells, and next to this will having a thin segment where it is made up of the squamous and epithelial cells. And here this descending lymph is mainly meant for the absorption of the sodium and potassium. It is mainly meant for the absorption of the sodium and ions are absorbed, that is sodium and potassium ions are absorbed in the descending lymph, and here there is no absorption of the water. In water it is not helpful for absorption. That is here descending lymph is mainly meant for the sodium and potassium ions are absorbed in this layer, in this segment. It is impermeable of water and helpful for absorption of sodium ions only. Impermeable of water, impermeable of water and permeable of the sodium ions. Next, coming to the second one, that is ascending lymph. So here the ascending lymph The initial part of the ascending lymph is made up of a thin segment. The initial part of the ascending lymph is a thin segment where it is made up of the squamous epithelial cells. It is made up of the squamous epithelial cells. Next to this, it is made up of a thick segment. It is made up of a thick segment where it is made up of a cuboidal and epithelial cells. So here in initial part of this lymph where it is thick segment which is made up of cuboidal, here will having the less amount of microvilli are presenting. So here these are opposite, that is in ascending lymph that is opposite to the ascending and a descending lymph. In a descending lymph, initial part is a thick segment and next to the thin segment which is made up of cuboidal and squamous. But in ascending lymph, here it is a thin segment later than the thick segment. Here it is made up of squamous and a cuboidal epithelial cells. And here function is here it is impermeable of sodium and potassium, impermeable of sodium and sodium ions, and permeable of the water, and it is a permeable of the water which is taken place. So here these are both are opposite to the one another. One is helpful for absorption of sodium and potassium ions. It does not helpful for permeable of water. And second one is it is permeable of water and impermeable of sodium ions, where these are opposite in directions are taken place, which is the longest part of the nephron, where it is an U-shaped structure where it consists of an descending lymph as well as the ascending lymph, which is made up of two segments, that is thick and thin segments which is made up of in descending lymph, and in ascending lymph it is made up of thin and thick segments which is made up of the squamous as well as the cuboidal and epithelial cells. Next, coming to the next one is a distal convoluted tubule. Coming to the distal convoluted tubule. So this distal convoluted tubule which is presenting, it is not having that means distal means end of the tubule, that end part of the nephron is called as end part of nephron, or which is far away from the nephron, the part which is far away from the nephron, and it is not having a much tube-like structures. So they are called as distal convoluted tubules, where here it is in 5 centimeters in length. It is 5 centimeters in length, and here this distal convoluted tubule which is presenting, here it consists of it is made up of a cuboidal, it is made up of a cuboidal epithelial cells, cuboidal and epithelial cells. Here there is no microvilli are not be presenting. There is no microvilli are not be presenting. It is made up of cuboidal cells, and a distal convoluted tubule is mainly meant for the, it is meant for tubular, it is meant for a tubular secretion. It is meant for a tubular secretion. It is meant for tubular secretion means what are the waste materials are secreted that are entered from peritubular capillaries into the nephric, that is into the peritubular capillaries into the distal convoluted tubule. That part will be entered from this, it enters into the collecting duct. From the collecting duct it passes to the urinary bladder. So here it is mainly meant for tubular secretion, that means they secretes the waste materials which are present in peritubular capillaries. So they are called as, it is meant for mainly for a tubular secretion which is made up of a cuboidal and epithelial cells. There is no microvilli are absenting, where it is in the 5 centimeters in length, and it is the last part of the nephron where it is called as end part, and also it is a distal, means it is a far away, it is a far away from the nephron, so it is called as a distal part of the nephron. So it is called as distal convoluted tubule. Next, coming to the collecting duct. A collecting duct. So what is this collecting duct? Means it is not part of the, it is not part of the nephron. It is not part of the nephron, but each nephron will connect. But each nephron will connect. But each nephron will connected to the, that means each nephron is connected to the collecting duct. Collecting duct, where here each nephron is connected to the a collecting duct, where it helpful for excretion of the waste materials, where here it is present in the straight tube which is presenting in the, it is presenting in the renal medulla. It is present in a renal medulla. It is present in a renal medulla, and proximal distal convoluted tubule is present in a renal capsule, renal cortex. It is present in a renal cortex, and the collecting duct is presenting in it is a straight tube which is presenting in the renal medulla, where it connects, the all nephrons are connected to the collecting duct, where it, in the name only indicating collecting, it collects all and unessential, that means the waste materials which are secreted by the kidneys that are collected by the collecting duct. So each nephron is connected to the collecting duct. So that is where it is now mainly made up of a columnar epithelial cells. It is made up of a columnar epithelial cells. It is mainly made up of a columnar epithelial cells, where each part is connected to the, each part of the nephron is each nephron is connected to the, it is not part, but each nephron is connected to the collecting duct, where it collects all the waste materials into from the nephrons, and also it is present in renal medulla, and it is made up of, it is straight connecting tubule, and it is consist of, it is made up of columnar and epithelial cells. Next, coming to the last one, that is a peritubular capillaries. The last one is the peritubular capillaries. Next, coming to the last one, the peritubular capillaries. Peritubular capillaries. Peritubular capillaries. So here it is a network of capillaries. It is a network of capillaries. It is a network of capillaries. Network of capillaries which are connected to different regions of nephron, where here it is connecting, that means here it is a group of capillaries, a network of capillaries. Network means many capillaries are called, that means this after leaving of an afferent renal arteriole, from an afferent renal arteriole, here this is connecting the different regions of the nephron, where here these peritubular capillaries are run parallel to the Henle's loop. Where runs to the parallel to the Henle's loop. So that peritubular capillaries which are run parallel to the Henle's loop, they are called as the vas recta. That means they are called as the peritubular capillaries. Capillaries which are running, which runs parallel to the parallel to Henle's loop, parallel to Henle's loop is called as the vas recta, is called as vas recta. So what are vas recta means? The peritubular capillaries. What are vas recta means? The peritubular capillaries which are running parallel to the Henle's loop, that is in between ascending, descending as well as ascending loop, they runs parallel to the one another, then which is arise from the an efferent arteriole. It runs, it passes, it connects the different regions of the nephron along with the Henle's loop. That Henle's loop is having the parallel to the one another. So that peritubular capillaries which are running parallel to the Henle's loop, they are called as the vas rectas. And this vas recta and the peritubular capillaries, the vas recta and peritubular capillaries are meant for the, they are meant for absorption of, reabsorption of, they are meant for a reabsorption of the essential elements from an essential elements from the nephric filtrate. These are helpful. That means peritubular capillaries as well as vas recta is mainly meant for the reabsorption of the essential elements which is presenting in the nephron. From the nephric filtrate, again from nephric filtrate, they secreted, that are absorbed and passes, and again from non-essential elements which are present from vas recta and peritubular capillaries that are secreted into the a distal convoluted tubule. So we can call that as a tubular secretion. From tubular, from the DCT, that is distal convoluted tubule, that passes to the collecting duct. From collecting duct, it passes to the urinary bladder. From urinary bladder through the urethra, these are sent outside through the anus. That waste materials are excreted and outside. So that what are that now in the next class we will discuss, that is how that is a urine formation, how urine formation is taken place, that we will discuss detailedly in the next class. But here mainly that peritubular capillaries and vas recta, peritubular and vas recta are mainly meant for the reabsorption of essential elements, that is absorption of essential elements is taken place in the peritubular capillaries as a vas recta. So these are the different parts which are presenting in the nephron. That is first one is Malpighian tubules, where it consists of the Bowman's glomerulus as well as Bowman's capsule together we can call as a Malpighian tubule. From the Malpighian tubule, it consists of proximal convoluted, from proximal to the Henle's loop, from Henle's loop to the distal convoluted tubule, from distal convoluted to the that is a collecting duct, from collecting duct to the peritubular capillaries. So this is the where the different parts which are presenting in the nephron, and each part having its own important which helpful for absorption of essential or non-essential elements that is taken place. So this is the different parts which are present in the nephron. Now coming to the last one, that is the functions of the kidneys. Coming to the last one, that is functions of the kidneys. So here main function of the kidney is it helpful for the maintenance of the homeostasis. The homeostasis are an internal body environment or internal environment. So here it helpful for the maintenance of the homeostasis. Homeostasis means it maintains a constant water balance in the body of an organism. So we can call as the homeostasis or internal environment. And also the main function here, the main function maintenance of the main function function of kidney is excretion. That means here the excretion of the waste material, that is the excretion means the elimination of the nitrogenous waste material such as urea, uric acid, ammonia that are excreted mainly, which are presenting that are excreted mainly the nitrogenous waste material, the toxic substances which are present in our body, that are main function of kidney is to eliminate the excretion, that is waste materials are excreted from our body. And also here it helpful for the maintenance of acid and base balanced in the body. That means excess acid and bases which are present in the body, they are eliminated or they are excreted outside from the body. That is, so here it maintains the acid base balance will be maintained by the body of an that is maintained by the kidneys. And also if you see in here the fourth one is it helpful for the ionic balance also. It helpful for the maintenance of ionic balance. It also helpful for the maintenance of ionic balance, and also here it helpful for the maintenance of the water balance. Balance, that means excess water which is presenting that excess water will be eliminated outside from the body, where it does not helpful for going on. That means it does not helpful to eliminate the waste material, that is excess water which is presenting, that is the excess water will be eliminated, and which is how much of water is required for the body, that will be maintained constantly, and a reabsorption of excess water which is presenting, that will be not eliminating, that is the reabsorption of water will be taken place. So it does not, it prevents the losing of water from our body, where here water is very essential element, where here we taken place. Here if we take 5 liters of body, 5 liters of water we are taken place per day. Here only 4.5 water it is absorbed, only half liter of water only we are eliminated in the form of urine. Remaining 4.5 liter of water is reabsorbed in our body. So that reabsorption is mainly taken place by the hormone called as an ADH hormone, antidiuretic hormone. Due to the presence of this antidiuretic hormone, ADH hormone, antidiuretic hormone, or otherwise you can call this as a vasopressin also. So vasopressin or ADH hormone is helpful for reabsorption of water in the body, where that is maintains in the body by the kidneys, and also it helpful for maintenance of ionic balance. The sodium, potassium, calcium, magnesium, all the ionic balance which are maintained, it will maintain excess ionic ions which are presenting, they are eliminated from our body, and also it maintains acid base balance in the body, and also the main function is excrete the waste material, that is metabolical waste which are produced from the cell, that are eliminated outside. Why? Means they are very toxic for the body, so they are eliminated from our body. That is the main function of the kidneys. And here it maintains the homeostasis, or internal environment will be maintained. So these are the various functions which is performed by the kidneys. So this is where that is called as, this is the part that is the structure of the nephron. So here nephrons are the basic, these are the structural and functional units of the kidneys. Why? Means they give a definite structure for the kidney, and also performs means it eliminate the excess of water or ions or acid bases or excrete waste materials from our body. So they are the functional, so their nephrons are the and structures of structural and functional units of the kidneys. They are also called as uniferous tubules. So in each kidney will consist of 1.5 millions of nephrons are presenting. So in the next class that will be discussed about how the urine formation is taken place. Now we have studied the structure of the nephron. So in a next class we will discuss how that is urine formation, how the urine is formation is taken place in the nephron from Bowman's capsule to the collecting duct, how it is excreted outside the waste materials, that will be discussed in the next class.